And here we are. And do you want to ask your question now?
I do.
Okay.
Okay.
So here we are with our first question of the day. Good morning, students.
Good morning.
So we talked about, well, in the video you talked about how DNA methylation affects ants, and specifically the queen ant, and how it was fed differently as a larva. Is DNA methylation affected similarly by diet in humans?
So the question is talking about DNA methylation, and the, DNA methylation comes up in some different kind of context or examples. And for us, the first one is this idea that when cells differentiate, they really typically methylate the genes that they're not going to use anymore. And we'll see. We want to understand that. And DNA methylation seems to always be involved in gene repression. And so it kind of makes sense that the muscle cells, as they differentiate, would be methylating genes that they're not going to use. Like the insulin gene is present, but the muscle doesn't want to express it. So that's our first thing that we think about when we think about DNA methylation. There are some other examples, and we'll learn about a couple of them, but our kind of take-home one for our course is, there's a lot of DNA methylation during differentiation. And that'll come up a few times in our course when we talk about stem cells, because stem cells are not differentiated, and they tend to be under-methylated on the DNA, right? So they can still express a lot of genes. And so we'll see that will be a relevant take-home message for us. And yeah, there are some other examples, and in the video, the question is about the ants and how their diet can affect DNA methylation. And the question is, does that happen in humans? And I think that there's evidence that diet affects DNA methylation in humans. I don't know that specifically, but in general, diet can affect DNA methylation in a lot of different animals. And so I kind of think it's probably been established that it could affect humans too. And yes, so the answer is I think so, and certainly in lots of animals. And so the anticipation would be that there'd be some examples in humans if they haven't been shown yet. Okay, so let's get started on our course, and super welcoming questions, and then we also need to have a sign-up sheet, which we're going to do like this. Okay. All right. And so this week, at the end of class today, we'll take about 20 minutes to do this quiz. And the quiz is based on the muscle differentiation clay model videos. And so we have time to ask questions about them. But the questions are going to be all from the videos like that. And then at the end of class on Thursday, we'll get out some clay and make some DNA clay models to, again, just be hammering those facts and concept home and terms like that, because we're just going to use them a lot. And this is, I consider, still intro bio level stuff, but there have been some good questions, and trying to understand this and really let things really sink in. And because it's a surprise, because it's physical stuff, and so you can understand it. And you've heard about stuff like genes and gene expression before, but we want to sink in so we can really use it, okay? And so that's the first thing. And so we'll have that. We're not going to have a concept chat today because we'll have a quiz. And if we get to 20 minutes and toward the end of class, and I'm not handing out the quiz, just remind me, you remember we're supposed to have a quiz, okay? Then on Thursday, Riley is going to visit, and he's going to be a tutor for our class, an informal tutor. He's just great, really nice, and he took the class last year. And when we think of Riley, we think of this CAR T-cell therapy. It's a kind of cancer treatment, and Riley is going to come to class 4 times and be explaining it to us. And so we're going to use Riley as a novel example of one concept in our course, and that is this certain type of cancer therapy. And it's all about molecular and cellular biology, so it's a really good example, and that's why we're going to do that. Then you should have read or be reading chapter 1. We'll start talking about chapter 1 today. I'm not gonna go through the chapters. I'll talk about complementary stuff, different stuff that's related, and so it'll be good for us to get into that. Some of the questions on the exams will be from the book that we never talked about in class. And so we want to find out how to navigate through the book and really understand it, and be able to, understand class as well. Most of the questions will come from class. Most of those will be from both class and the book, like that. And so we'll be getting into that. And then by the end of this week, you should start reading chapter 4. We're never going to read chapters 2 and 3. Chapter 2 is about biochemistry, and you can take biochemistry if you want. So it's about metabolism, it's about molecular structure, and metabolic reactions and stuff like that. And you had some of that in intro bio, but you could also, could pick it up in other courses. And that's part of molecular biology, but it's not part that we're going to emphasize. And then chapter 3, I forgot, or chapter 3 is that, then chapter 2, what the heck? Oh, chapter 2 is like intro, and then chapter 3 is metabolism, like intro chemistry and metabolism. And that's just not going to be a huge focus in our course. It's super important stuff that, you should get from other courses. So we're going to go from chapter 1 to chapter 4. Chapter 4 is called Fundamentals of Molecular Biology. So chapter 1 that we'll talk about is a big overview, and then chapter 4 is still an overview, but starting to dive in, and it's starting to dive into really our author's career, which he's a little bit older than me, and it's like all this stuff has happened, and these people are still alive. Jim Watson, the co-discoverer of the structure of DNA, he's still living. You can call him up like that, say, I'm struggling in this class, okay? So that's what we're doing in our course. And so today, and let me just show you this. This is an email. You don't need to scramble or write it down. It's from what's called the Learning Commons or the Tutoring Center. And although Riley's going to be our specialized tutor, he understands this material. Over at the Learning Center, nobody understands this, right? It's an advanced course, and they won't be able to help you with the specific content. But the Learning Center is really great for things like time management and study skills and how to tackle assignments and things like that. So it's a really good resource, and you've got emails like this already about welcome, and here's the Learning Center, and it's fantastic. It's fantastic. I grew up in a little farming town. The high school was really weak, and I wish I would have had resources like when I showed up at college to learn how to learn, because a lot of students like me were not challenged in high school and really need to learn how to learn in college, how to best learn, really, to sit down and focus and study productively. And so, so take advantage of that. One example I'd already talked about is strategy for learning in our course is all the exams are going to be 2-point questions. So it is a kind of flashcard knowledge. Like, it's not multiple choice, it's stuff you have to know. Like, here's a term, what does it mean? Like that, right? Okay? And so flashcards are good for this kind of learning. A step up from flashcards is to do something like this. This is an image, right? It's all physical, it's all stuff you could see, and you could draw a simple big image like that, and you could put flashcards on the different spots on the image. So instead of just having a stack of flashcards, they're actually laid out. You can pick them up, but you can set them down, and you can tell the story, right? You can start out with an unlabeled drawing of this or any of many things we're going to have this semester, and you could lay flashcards out as you talk through it like that, right? And then you'll be able to point and talk, and that's a really good way to stack up learning, is to tell a story. It's a physical story, or it's a timeline type story like that, okay? And so that's that, and we'll have a quiz at the end of class today. So do people have questions about that video stuff at all? Yep.
You had said in one of the videos that I think it was methylation was an exception to like changing the DNA sequencing. Do you mind just like going over why that is briefly?
Yeah, so the first thing to say is I like to say that the DNA is not changing. And when scientists say that, as we develop and grow and live our lives, the DNA is not changing. We're referring to the AGC letters, like that. That DNA sequence is not changing. And yes, there could be some mutations, right? They can cause problems in our lives like that, right? But in general, we're born with the same DNA sequence that we die with, like that. And that's true in general. That the DNA is a storage molecule and it's pretty steady, like that. And so I like to say that a lot. Oh, the DNA is not changing. There's one exception to that, and that is that sometimes the DNA can have a methyl group added to the C's, the cytosines, like that. And when that happens, the role for that appears to be that it will cause repression of gene expression, like that. Yep. Other questions people have about that? And it could be anything at all, like structure of the course, expectations, or maybe you're already in chapter 1. But yeah, since we're doing that quiz, also that. Questions about that? Yep.
Has there been any link between like abnormal methylation and cancer?
Has there been any link between abnormal methylation and cancer? Almost for sure. Yeah. I'm just trying to think of a specific example, like that. I'll just say as a sidelight that before I got a job here, I worked in a building at the University of Washington in Seattle that was tall, so we rode the elevator a lot, and there was a graduate student named Mary. And sometimes I'd ride the elevator with Mary, and Mary was never happy. Mary was always frowning, like that. And I grew up, again, in this little farm town, so I say hi to everybody. Hi, Mary. She'd get in the elevator. Hi, Mary, like that, She'd be frowning, she wouldn't answer, And I didn't do it to tease her, it was just like, that's, hi, Mary, and she wouldn't answer. One time, she had a novel that she was reading under her arm, and I said, oh, hi, Mary, what's that you're reading? And she said, it's a novel about the future when they clone out of the human race, they clone out and get rid of the Y chromosome. I was like, oh, it's like, that's me. Talking about me, right? And then the only other time Mary's ever said anything to me, she got in the elevator once, we're riding up, and Mary said, she just says out loud, she says, you have male answer syndrome. And I'd never heard of it, but you've probably heard of it now. And that is that, like if you ask, a kind of stereotype is you ask a male a question, they answer, even if they don't know, they just answer. And when I learned that, it was just such a release, because now I can just say, I don't know, right? I don't know. And I feel like I'm doing a good job. I don't know. DNA methylation and cancer? I don't know. Right? I don't know everything. I don't even know how to get to them all. So I don't know, but I'll say almost certainly there's some story out there. I just can't think of if I know of a specific one right now. Yep. I'm sorry, but yeah. So sometimes I'm like, I don't know. Whatever. I don't get it. I've tried careful not to speculate or to say stuff that I'm not sure of. So, yep. Yeah. Other questions people have? Yep. Yeah. Yeah, just go for it. Yeah.
So you talked in video number 3 about muscle differentiation after birth. So we were talking about satellite cells. So after a satellite cell gets stimulated from muscle strain or whatever, are the steps to get to a myoblast, like what are those? Like are they the same or like?
Yes.
What happens there?
Okay. So it's a very nice thing that'll come up. Later in our course, we'll talk some about stem cells. In chapter 19, we'll talk about stem cells. And these satellite stem cells are a good example. And the stem cell story is really important in molecular biology, so it's going to be an example that will carry through. And satellite stem cells are called adult stem cells, and that's kind of a misnomer because they're present through our whole life. They're present in a fetus or in a baby, in a child, in a teen, an adult. And we have some different satellite stem cells. We have different adult stem cells in different locations. We have these satellite stem cells aligning our muscles. We have stem cells in the bone marrow, like that. And in adult stem cell, here's a stem cell, and it divides like that, and it's said to be self-renewing. so as it divides, it's going to produce another stem cell. So the population of our satellite stem cells is not decreasing, it's maintained. Okay? And then it divides asymmetrically. It's said to divide asymmetrically. And so it has two products. One, the self-renewing stem cell, and the other is just generally called a precursor cell. And it's that cell that's going to go on to form a myoblast, and then that's going to differentiate to a muscle. And so that happens anytime the satellite stem cells are stimulated. And they're stimulated when we're growing as children and our muscles need to get bigger, because our muscle fibers can only increase a little bit by increasing the size of the cells. And so as we grow as children, we need to add more muscle cells. But the muscle fibers are differentiated and they're no longer dividing. So we need to have a new source of precursors to make more muscle cells as kids. Then throughout our life, we might get injured and our muscles can heal. And we also can work out, or some animals migrate and their muscles increase. Our muscles can increase in mass due to environmental factors, such as in case of birds and migrating birds, the day length. In case of us, working out, getting bigger muscles, right? So and those are all examples of that, where those came into play. And so that's a story, and this will become a really nice question for us later in the semester, when we're like, well, how are these cells becoming different? How is that cell moving down that pathway to become a muscle cell? It's the adult stem cells have limited potential. They can only form certain types of cells, and the satellite stem cell can make muscle cells. So it's set up to be held in reserve throughout our life, ready anytime we need more muscles, like that. Yep. And other organisms have them. I think all vertebrates have muscle satellite stem cells, like that. Yep. Other questions? Yeah.
So you talked about the coding region that is used for transcription. You mean like a coding strand that goes with the template strand? Is that what you're talking about?
Yeah. And so let's just look at that in more detail. And so let's go with it this way. Let's start out and say, let's just define gene. Okay, so we'll start with the definition of gene and we'll work our way to that question. So a gene, that's from the field of genetics. The field of genetics is only 120 years old. And for the first half, almost the first half of it, they didn't know what a gene was. So it was theoretical, right? It's, kind of, we know it exists, but we need to start working on it, right? And it's surprising, right? Because genetics, it just sounds like that. It's been here forever, but it's not. Genetics is a young field in science. And so the first thing is we're going to define gene, and that could be a question on the exam, right? Definition of gene. And the examples of questions on the exam questions always have really like crisp, direct answers. And if you know that, great. But then we'll talk about them and explain them more. And so almost all the time students have longer answers on the exam because they'll be explaining it like we learned in class. And that's fine, like that. Let me just say that after you take an exam, what I do is right after I grade them, I make a video of myself taking the exam. And I talk through the answers, and I talk about some of the wrong answers, that I just read, like that. And everybody watches that, and I don't name names or anything, but everybody gets a feeling for that. And one thing I like about it is when I make the answer key video, I talk about sort of the direct answers, like that, right? So to get us back to that again, that it's like boom, like, so here comes a pretty direct answer. Okay, definition for the gene. And it's interesting because, like, you can ask other, professors, hey, what do you think about a definition of gene? And they're like, oh, that's hard. And it's because it's kind of used to be theoretical. It's not theoretical at all, right? So the first thing you want to say about a gene is it's physical. Always. That's the heart and soul of our course, is it's a physical thing we're talking about. This stuff is very physical. Biology is called a natural science, right? That's historic. It should be a physical science now. It is physical. It's maybe more physical than physics, right? It is just rock hard physical stuff, right? So it's physical, and it's composed of DNA. Okay, and then it's got these two parts. Now, we just want to say this is a universal definition of gene, so we have to be careful with some of the terminology because, especially in something like microbiology, they use some different terms for the same things, right? We're talking about physical stuff, and they point to it, and they just use a different term. Like instead of saying gene, they might say operon, like that, you know, and you're like, you know? Okay, so here's the DNA, and it's made of two parts. And one part is going to get transcribed, and so we call it the transcription unit. And so we'll just show that's the part that's going to get transcribed, and that's the part we're going to zoom in on, right? Because we talked last week about how in these two parts, there are different stories. And we have a question about the coding region, so we're going to zoom in on that. Because that's going to be the part that's the output, like that. And so one is the transcription unit, two parts. One is the transcription unit, and that's got the information for the output, which is going to be RNA. And then the other region is the regulatory region, and I like to just draw a dotted line because the regulatory region can cover a lot of territory, like that. So the second one is this regulatory region. And that's a good, and let's put in that word cis, right? That means physically connected, because that's part of the definition. It's physically connected. So cis regulatory region, right? We learned about that. It's not over there, it's not the MyoD gene, it's the myogenic gene. It's that gene, and it's got to be cis. It's got to be physically connected. So that's really important to have that. And then the last thing you just say in a definition about a gene is that it affects the phenotype. So it's part of the genotype, and it affects the phenotype. And that's a really great definition of a gene. It's really rock solid. We haven't put in those fuzzy terms, right, like that, but it's good. So let's just add a couple of those terms further, but that's a good definition of a gene. Does anybody have any questions about that? So it's physical, composed of DNA, made of two parts, the transcription unit, which is the output, it's going to get transcribed, and the regulatory region.
You said something before. You said that it affects the phenotype. It's a genotype that affects the phenotype. Is that what you said?
Yeah, it's genotype. This is the genotype, and it affects the phenotype. So that's part of what a gene needs to do, is it needs to affect the phenotype. Just as a side, like I'll just mention, there's some broken genes in our genome. They're called pseudogenes. They don't affect the phenotype. We wouldn't call those genes, they're pseudogenes. So just, to really pin it down, is it's got to play a role in the phenotype, like that. And then I'm just going to draw a box around that cis word, and I think that's a really good word for us. It's an old-time genetics word. You don't hear it very much, but it really helps us go, yeah, no, we got to have that thing tethered, physically tethered, like that. Some of these DNA sequences, the letters don't matter. It's just that its strand is got to be tied, connected like that, okay? But let's just add some of these fuzzy terms. The first one is, and they're not all fuzzy, but just to say, we like to talk about this region here of the regulatory region. We like to call that the promoter. I'm just going to add a word that is a term that a lot of molecular biologists use. They like to call it the proximal promoter. Proximal means really close or nearby. And that's a good term because the promoter is always right next to the transcription unit. The transcription unit starts at the nucleotide called +1, and the promoter is right next to it. Then we have this much more interesting part, and it's the enhancer or multiple enhancers that are further away. And that's interesting because that's a binding site for some really interesting dynamic transcription factors that we'll learn a lot about. Like MyoD binds the enhancer of the myogenic DNA. And so those would be all terms about DNA, right? The enhancer is made of DNA, composed of DNA. The promoter, composed of DNA. The transcription unit, they're DNA terms, and they're parts of the gene, okay? Then over here, then we get closer to the question, and we say, well, there's actually two classes of genes. One we call coding genes that are going to code for proteins, and the other non-coding genes. And we just need to have that in our explanation and understanding because we're going to talk about both during the semester. So coding genes are genes that code for proteins. Most of the time we learn about those because they make interesting proteins. The product of a coding gene, I'm just going to erase this now, and we'll get into this a little bit. You know, so we're looking at double-stranded DNA like that, okay? And we're focusing in on the transcription unit. And we have coding genes that are they code for proteins. And so those genes are going to get transcribed into RNA. And that RNA is called messenger RNA, mRNA. It's the output of the DNA, but then that output leads to synthesis of a protein. So there's a ribosome, and in all cells on our planet, the protein synthesis takes place in a ribosome, and the ribosomes use the mRNA template to code for the amino acids in every protein. A really important concept is we're humans primarily because we synthesize human protein. A dog is a dog primarily because it synthesized dog proteins using the dog genes that it inherited from its parents. Like that, right? We weren't here. How did we get here? How did we get here as a human and not a maple tree? And the answer is, well, we have human genes. And the most important ones are our coding genes that code for all human proteins. We might eat insulin in our diet, but we'd never use it, even if we were really diabetic. We just degrade, we metabolize proteins in our diet to individual amino acids, so each of our cells can synthesize the proteins they need, and they're human proteins, like that. Okay? Okay, so in this coding gene, so we're focusing on coding genes, so coding gene. You know, and this could be any of them, right? Insulin, hemoglobin, myogenin, you know, all of the coding proteins. So within that, there's a coding region. So a portion of the transcription unit, a portion of the output, is called the coding region. And the coding region is made up of codons, which are triple letter
Base pairs?
nucleotides. They're not base pairs, just triple letters that each code for one amino acid. So the coding region is these triple letters, nucleotides, that are used by the ribosome to tell, oh, this codon should be methionine, and then alanine, and then glutamine, and so forth. Yep?
Is there like another scientific word for that? Like the UTC read?
Well, you use a couple of terms, and we're almost to get to, I think, the terms you're referring to, okay? So coding region, like that, okay? And now to address this, I think, your question, we're going to zoom in on that coding region, like that, okay? So now I'm just going to draw that DNA, and it's going to be the DNA within the coding region, and we are going to untwist the DNA, double-stranded DNA in that region, okay? And it's going to have to, we're going to have RNA polymerase come in, so that's RNA polymerase that's going to transcribe the gene during gene expression into RNA. And the RNA polymerase uses one of the strands. So let's say it uses this strand, and it transcribes it to make the complementary strand. And so let's just put some letters on that so you see what's going on there. Let's just say that this is going to be like an A and a G and then, you know, 3 C's. So the corresponding DNA strand then would be a T and a C and 3 G's, right? So complementary strand like that, right? It's transcribing that, it's copying the DNA complementary like that, okay? The next thing is to say is that there's an orientation of the DNA, and we'll talk about that in some more detail. There's an orientation of the DNA where, for any given strand, there's a 5' end and a 3' end, like that, okay? And the DNA is said to be anti-parallel, and that means that 2 DNA strands are built in opposite directions. So, if this is 5' and 3', then in that DNA strand, this must be 5' here and 3' here. And then down here, this other strand that we're not focused on, that must be 5' and 3'. Okay, like that. All right. So then the next thing to say is just to mention that for this enzyme RNA polymerase, RNA polymerases on our planet can only go in the 5' to 3' direction. They can only synthesize RNA in that 5' to 3' direction of all of us, all the cells on Earth. And all the cells on Earth have to transcribe genes to the RNA, and all RNA polymerases are always going in the same way. Just a little side note, because it's a different topic, but DNA gets replicated, that's a different topic, by DNA polymerase. And all the DNA polymerases on our planet can only synthesize DNA in that 5' to 3' orientation. Collectively, RNA polymerases and DNA polymerases are called nucleic acid polymerases, and all of them go 5' to 3' on our planet. So the convention is just when you see it written out, letters like this are always written in the 5' to 3' orientation. Okay, then the next thing to say about that is, see how that RNA, right, that's the messenger RNA. So that's going to be used by the ribosome. And it's going to read that AGC as a codon, and it's going to tell it the amino acid, right? And so that's called the messenger because it's bringing it to the ribosome. That strand is also called the coding strand because it has the code, okay? The mRNA has the code, it has the message. So I think that's one of the terms that, Hannah, you were asking out, like that, okay? So that's often called the coding strand, okay? Then,
so the complementary strand, when it's taken up by the ribosome, becomes the coding strand?
this strand
Yeah.
is the strand that's going to be used by the ribosome.
Yep.
And so that's the actual letters that are going to be used by the ribosome, so it's referred to as the coding strand, like that, okay?
But when it's in the RNA polymerase, it's the complementary strand because it's complementing the DNA?
Yeah, so you could call this is complementary to that DNA, like that, okay? All right, so then that coding strand, that coding term, that's not so common. Here's the more common term, and that is that this strand makes sense to the ribosome. This strand of the DNA is called the template that RNA transcribe uses, and it would not make sense. It's the complementary strands. If you look, though, down here, this strand that we haven't been talking about, this strand actually has that same sequence as the RNA does, right? And so this strand is referred to in the DNA as the sense strand, like that. And then this strand, that's the template that's used by RNA polymerase, is called the antisense strand. So DNA is double-stranded, and in the transcription unit, there's the coding region. And of the DNA in the coding region, there's the sense strand of the DNA and the antisense strand of the DNA. The antisense strand of the DNA is used as the template by RNA polymerase to transcribe the DNA. Yep. And yes.
Not to interrupt, I'm just going back.
Yeah, this is great. Always a good time to interrupt. Yeah.
For the RNA polymerase, you were saying it goes from
5' to 3' in that direction. Like it,
okay.
yeah.
And when you're saying it's going from, that's when you're creating the complementary. So it's actually like reading 3 to 5, but creating 5 to 3.
Yeah.
Okay.
Yep. Yeah, okay, and so this was getting back to your original question, and is that what you were asking? That gets help,
Yeah, no, I just.
the terms and concepts? And that's all in the coding region, right? And it's in the transcription unit, and remember that that's the product. So anytime we talk about a mutation where the product is altered, the mutation must be in the transcription unit. Sickle cell anemia, people have sickle cell anemia, they inherited a mutation in the DNA where the hemoglobin protein is sticky, and it sticks to the hemoglobin. Where's that mutation? Oh, it's in the coding region. You know, like that, yeah?
What do you call the parts of the coding, like the size of that?
Yeah, the part of the transcription unit that's not translated is referred. Yep. So I'll draw that out. Yep. So just to draw the DNA. So here's some DNA. And it's a coding gene, and so we've got the transcription unit there I've drawn, and then there's the coding region, and the coding region is just a part of the transcription unit. And the unused part of the transcription unit there, that's called the 5' untranslated region. Translation is what the ribosome does. So the ribosome gets this messier RNA and it says, like, that's not useful. Oh, here's the coding region, I'll use that. So it's called the 5' untrans because it's at that 5' end of the gene. The way we draw it is always that's the 5' end. And then this is referred to as the 3' untranslated region. Like that. Yep. Okay, other questions people have about stuff?
Going back to the video, we talked about MyoD binding to the enhancer, and then transcription proteins binding to the promoter. Is the MyoD and the transcription proteins bound together? And that's what the regulatory region making that tether is for? And then that connection signals the RNA polymerase to start doing its thing?
Yes. So the function of all transcription factors is to regulate the rate of transcription. Some transcription factors activate transcription and increase the rate, some decrease the rate. So somehow transcription factors regulate the rate that the RNA polymerase lands and goes down transcribes the gene. When a gene is being expressed, like insulin in our pancreas, the RNA is unstable, and so the gene needs to be continuously expressed. And so anytime a gene is expressed, like myogenin, it means that RNA polymerases are pretty consistently landing on the gene and transcribing it and producing a product, like a little assembly line like that, or a busy airport where the RNA polymerase is landing at the airport like that. And they go down the runway and then they just take off again when they finish transcribing, like that. Yep. And so how do these transcription factors do their job? And it turns out that the order we're reading chapters is, we're doing chapter 1, then 4, fundamentals of microbiology kind of intro, and then we're going to go to 9. And 9 is where our book deals with this. And you'll see when we get to 9 that there's a lot of other additional stuff that the book wants to talk about. And we'll be happy that we're driving forward with these videos and the physical nature of this, because you just would lose it so fast that it's a physical, commonsensical, matter-of-fact story like that. In that process, at chapter 9, we get to it, we'll talk about some actual molecular mechanisms for how transcription factors do that job. For right now, let me just show you this. Let me just show you this right now. Here's a transcription factor, and it's upstream of the gene. Here's the enhancer, so that enhancer's DNA. And this transcription factor is bound to the DNA. So this transcription factor is a protein, and it's got some shape to it, and part of it is called the DNA binding domain. And you can think of it as a hand, like the transcription factor is a little bit like a shape, like a blob, and it's got a hand that can grab hold of the DNA, and it grabs hold of the enhancer. It grabs hold of the specific sequence of the enhancer. Enhancers are typically about 20 nucleotide letters long, and the hand just fits that and wraps around it with high affinity. And then what Jeremy's question is about is this transcription vector then must have another region like that, and it's called the transacting domain. And that's the part that's going to do the function of whatever it is, like somehow affect the rate of RNA polymerase. And we'll learn there's some real typical ways it does that, but it'll be satisfying. It'll be chapter 9 in a little bit. For now, just learn this, right? Most people are not molecular biologists, right? And therefore, we're just impatient. We honk our horn at them. Aren't you a molecular biologist? Get out of the way. Most people think that's it's transcription acting domain, but it's not. It's trans, because it's that genetic term again, where this is not cis. This is coming from somewhere else, and it's acting in trans, genetic. It's a genetic term like that. It's some other thing over there that could have a mutation, right? We talked about that. You could get no muscles because you could have mutations in myogenin, in the coding region, defective myogenin protein. In the enhancer, MyoD fails to bind and activate myogenin. Or over there in trans in the MyoD gene, and we don't make MyoD. Mutation in MyoD is just as bad as a mutation in myogenin, like that. So transacting. So it's got to have that, and that's the thing that Jeremy's asking about. What's that do, right? And it does some really cool things like that, where, like, I don't know. I'm almost always kind of almost choking up, but sometimes, like I say, here are the 3 main ways that transcription factors regulate transcription, and I'm like, they're so cool, because they're physical. They're tethered and they're doing this thing, and it's nuts. Yeah.
And then those.
But we'll get to that. But yes.
Transcription proteins stay bound to the promoter enhancer while the RNA polymerase is zipping along, and then releases, and then comes back.
Yeah. So these proteins are binding through non-covalent interactions that are specific and high affinity, but they randomly let go. But in general, when a gene is being expressed, these proteins, these transcription factors, are staying bound and are tracking one RNA polymerase and then another and then another, like that. Yep. Okay, other questions people have? Okay, and we can interrupt. We're doing great. Oh man, we are just doing great. There's so much to cover in this course. All right, so back to this, some questions, but then this is still structural stuff. I just want to remind you of these features of our course. Remember, you have to participate in class. That does not mean ask questions. It just means to either attend or take notes every time. I'm recording the class, and you'll have a week to take notes if you miss it, and email me pics. And you just have to do that, or you could flunk. You're not graded, you're just required. And that's described in the syllabus, and we just need to be solid. I don't suppose there are any other courses that have that. Then you also have to try to attend. So that's described in the syllabus. You just make sure you're square on that, because you're not going to get graded on it, you're just required, like that. And it could hurt your grade if you don't participate. Again, it's not participate like question. It means attend or take notes, and then also try to attend. And then most of the time we'll have these concept chats, so be taking notes so you have something to refer to when we say, what are the 3 important things that you think we talked about so far? And then we're going to have quizzes and assignments in class. When we have them in class, they'll be announced well ahead of time, and there's just no makeup. If you happen to miss, it's just, oh, you missed. And so, your car might break down and you just missed it, right? Like that. We'll have several of these, and the goal is to try to attend class, and if you miss one, bummer, but we're just going to roll with that strategy, okay? So that's just the structure. Then there's an option on the syllabus to do the 4-H STEM Ambassador Program, and you jump ahead with that on your own. I've described that, and you contact Sarah Sparks, Her contact is on our syllabus, which is on Brightspace, so you can find the syllabus on Brightspace and find the link to learn about it and jump on that. If you want to do it, I recommend it. It's a good experience. You can put it on your resume. It's a good personal experience, and it's interesting teaching experience of some grade school students. If you didn't hear me talk about it, take a look at it. If you have questions about it, let me know. But it's really a thing you just go for. And you have to decide by February 7th. That's the first training date. And so you'll have to decide, in the next couple of weeks. Okay, great. All right. Oop, and I got this a little bit out of place. How'd that happen? Definition of dean. Look, I was planning to talk about that, you guys. See how I'm doing my job? All right. So here's pictures from your book about chapter 1. There are 3 sections of chapter 1. Origin and evolution of cells. Like that. And so our authors want to talk about the first cell on our planet. And that's a really interesting thing. We're not going to talk about it at all. You have to read that, learn about it, and it's just not a big deal for us in our course. We have a different way to approach that. Then we're going to talk about some model organisms like mice and fruit flies and so forth. And we're going to be using those model organisms, so the author wants to introduce the concept of model organism approach. And you have heard of the concept of model organisms. Let's just pick fruit flies. Scientists love to study fruit flies, and there's some reasons they list, right? They say fruit flies are great to study because what? They grow fast. It's easy to grow them in the lab, right? Like that. And we already know a lot about them, okay? There's a reason that's rarely mentioned, and that is their genes are a lot like ours. And so we're going to talk about that. That was going to be our focus. So if it's ever anything about a model organism, the first thing you would say is their genes are a lot like ours. Fruit flies' genes are a lot like ours. Right? Not identical, because they're not human. They're a lot like ours. And so we're going to emphasize that. And so, there's kind of a breezy thing. We'll learn about that some, but they have genes that are a lot like us. Okay? Then the third section is about some tools like that. We're also not going to talk about those because we'll try to learn about some tools as we use them during the semester. But you're required to read about it, and some of the tools are hard, like super resolution microscopy. But that would be a good question for the exam that we might not talk about. There's a whole section about fluorescence resonance activated fluorescence resonance activated transfer, I guess. What's that, right? So there are sections. So it's good to say I have a one flashcard. We didn't even talk about it in class, but one flashcard about FRET or one flashcard about super resolution microscopy, like that. Okay. All right, so what is it that we are going to talk about? So here's what we're going to talk about, and we were dealing with this in lab last week, so some students in lab will be familiar with what we talked at the end of class on Thursday, and that is, we're talking about those muscle differentiation videos, and I mentioned before, every animal on our planet that has muscle differentiation occur in the same way. So it has a myogenin gene, like that, okay? And the genes have all been, lots and lots of genes have been sequenced. So the DNA's letters have been read and the information stored, and we can make comparisons. And so in the lab class, we just used our example. We said, hey, let's take the myogenin protein from humans and see if the computer can find the myogenin protein in dogs. They have muscles, right? And it's not surprising, they're mammals, they're closely related to us. It's not surprising that the computer could search for a match to find all the dog genes to see which one is most similar to us, and say that I think that's the myogenin gene, okay? And that search is a lot like a Google search, where the computer searches them and it's going to rank them. It's like, this is the best dog gene. Here's one that's not as good. Here's one that's even less good. but that best one is going to match really well. And that search program is called BLAST. So instead of Googling stuff, we blast stuff. And so let me write that down. And it's just a really simple computer program. It's like a word search on your computer too, right? You wrote a paper and you want to, I know I used the word library and I might have misspelled it. So you just search for library and the computer finds like that, right? It's just lining them up. So it's called BLAST. And it's actually officially has an exclamation point because it was written a long time ago and it's like only like 250 lines of code or something really famous where you're like, how come that coding person never won a Nobel Prize? Because it's just a really amazing thing. Because it's such a short program, it can do it really, really fast, right? And what it's essentially doing is it's taking what's called the query sequence, and these are just amino acids in the human myogenin protein. And it's just looking through the dog genome to say, hey, can I find the same amino acid in the dog genome? Oh, and then I found it, and then how about this one? And then how about this one? And it's just lining up those matches, and it's going through the whole genome, looking, and making longer matches, so it finds the longest, best match. And for each of these positions, it's kind of like flipping a coin, except for it's amino acids, and there are 20 amino acids. So to get this first match, it's 1 out of 20, and then 1 out of 20, and 1 out of 20. So to get 2 in a row match like that is a 1 in 400 chance. It's rare. But the computer finds lots of them because there's lots of genes like that. And then whatever, or it's, did I say it's 20 times 20, 1 in 400? No, what is that? 400. 400. And then the next one is 8,000, right? So when it starts finding matches, it can find really good matches because the proteins are typically a few hundred amino acids long. Okay? And the BLAST algorithm ranks them, and then it tells us the results. And it says, here's the best match. And then it says, what's the likelihood to find such a good match by just chance? Right? Like we flipped a coin, and we flipped another coin, and they all match. We picked an amino acid, we picked them, they all match. And because instead of flipping a coin with a 50% probability, we're picking amino acids with a 1 in 20, 5% probability, and we have lots of them, the matches and the probability that they would occur by chance are fantastic, right? And that's a key concept. And so I just want to show you this, and we'll have our quiz in a minute. And you don't have to see this, but this is the result, and it's ranked them. So it takes the human myogenin amino acid sequence and it finds it in dogs. It says, oh, I found a really good match. And over here it shows the percent identity. It says 80% of the amino acids are identical. That's not surprising because we're so closely related to dogs. And then it gives this probability. The likelihood that that match would be that way by chance is not 1 in 1,000 or 1,000,000 or 1,000,000,000, which is 10 to the 9th. It's 1 in 10 to the 115th. And it's like, you probably couldn't find anything else that scientists have evidence for that has that high of, or rate of probability. You know, what is the chance that these occurred just by random? And the answer is never in our universe. I don't care how big the universe is, it never happened by chance. When we see that, we say, oh wow, the interpretation is that the dog and the human shared a common ancestral form that already had myogenin. And we call that an ortholog. So we have orthologs of myogenin in animals that have muscles. And we have a slide with that image on there. There it is. So an important concept for our course is orthologs. Orthologs are 2 genes in different species that share a common ancestral gene. That's understood. It's theoretical. We don't know that common ancestor, but based just on that evidence, the statistic evidence from the DNA alignment, and then the study, and saying, well, when you look at what myogenin does in the dog, it's doing the same thing. It's expressed in muscle precursors and it stimulates differentiation, right? Combine that structure and function story to say, yeah, what do you think is going on? It's pretty obvious, right? So ortholaw, very strict definition like that. It's understood they share a common ancestral gene. So that's the key thing about model organisms. Why is a fruit fly a model organism? Lots of orthologs. Okay? And we have to stop and have our quiz, but we'll start with this on Thursday. And you'll see that it's a way we're talking about chapter 1, because we're talking about that evolution story, but from a different angle, a different feature than what the book chooses. Okay? So you can put away your stuff. When we finish the quiz, you can just head out. Just hand it in, you head out, and we'll do this again on Thursday. And